Welcome to Mr. G's Sleepy Science Channel. I'm your host, Mr. G. After 30 years of teaching various high school science classes, I realized that my voice apparently resonates at the perfect tone for putting people right to sleep. Hopefully my vocal superpower will work for you. On this episode, I will be talking about DNA, or deoxyribonucleic acid. DNA controls the production of proteins within the cell that form the structural units of cells and control all the chemical processes within the cell. First, let's talk about the structure of DNA. DNA structure, known as the double helix, was discovered in the 1950s by James Watson and Francis Crick with some help from Dr. Rosalind Franklin. And the basic double helix form has two long strands, each of which is a chain of nucleotide monomers. It is estimated that the DNA strand in the average human being is anywhere from 2 to 3 meters long. Each nucleotide that repeats over and over again in the double helix has three main parts. The first is a 5-carbon sugar, and since this is deoxyribonucleic acid, that sugar is deoxyribose. The second part is a phosphate group, and the third is the nitrogen base. And the nitrogen base makes the rungs of this double helix ladder, and the nitrogen bases can be one of 4 different kinds. There is adenine, thymine, cytosine, and guanine. And we mentioned earlier that the DNA structure are repeating nucleotides, so let's talk about the definition of a nucleotide. It is a segment of DNA made of 1 sugar, 1 phosphate, and 1 nitrogen base. And those nucleotides will repeat over and over and over again in the double helix DNA molecule. There are 2 different groups of nitrogen bases, and they are called purines and pyrimidines. The 2 kinds of purines would be adenine and guanine, and thymine and cytosine make up the 2 pyrimidines. So let's get back to the shape of the DNA molecule. Again, is described as a double helix. Helix, of course, meaning twisted around a central axis, and double indicates that the helix is 2-sided. So I would often tell my students, imagine if you took a ladder and were to twist it. That would give you a good idea of the shape of the double helix. So by definition, we say that it is 2 strands of DNA that twist around a central axis, forming a spiral structure. Again, discovered by James Watson and Francis Crick in the 1950s, along with some work by Rosalind Franklin. It would appear to look like a twisted ladder when the sides are alternating sugar and phosphate units. Again, 2/3 of that nucleotide. And so the sides are just going to repeat sugar, phosphate, and the nitrogen bases will reach across and would represent the rungs of that ladder. Another key or important nucleic acid found in the cell is RNA, and RNA stands for ribonucleic acid. RNA is made from DNA. Its primary function is in the synthesis of proteins. There are 3 main types of RNA. There is mRNA, and the M stands for messenger. Its job is to go in and copy the DNA molecule inside the nucleus so that its message can be brought outside of the nucleus for the manufacture of proteins. There is tRNA, and the T is for transfer, which is a single strand of RNA that folds back on itself in what is said to be a hairpin fashion. It allows for bases to pair of 20 or more kinds that each bond to only one specific amino acid. And then there is rRNA, or ribosomal RNA. It's found in the ribosomes, and at this time is not totally fully understood what its job is inside the cell. And now we're going to talk about the three differences that are between DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. First and foremost, where DNA is a double strand, RNA is only a single strand. RNA uses the sugar ribose in its backbone, and of course DNA uses the sugar deoxyribose as its 5-carbon sugar. And RNA uses the nitrogen base uracil in the place of thymine. So the 4 nitrogen bases used by DNA would be adenine, thymine, cytosine, and guanine, and in RNA, the nitrogen bases used would be adenine, uracil, cytosine, and guanine. Alright, and we're going to revisit where those different types of RNA will come into play here in a few minutes. Right now, we want to talk about DNA replication. If we go back to the cell cycle, this would be the S phase of the cell cycle, short for synthesis, and this is where the cell will make a copy of all of its DNA. And this is done in the process known as DNA replication. During this process, the 2 strands of DNA will separate at the bonds between the nitrogen bases, or is what is described as the molecule will unzip. Then new nitrogen bases bond with the old ones, forming 2 new complete DNA molecules. It will maintain the same relationship between the nitrogen bases thymine and adenine and cytosine to guanine, so an accurate copy is made. So this simply means when the molecules unzip, that will expose a thymine molecule and a new adenine will bond to it. And of course, conversely, on the other side, the free adenine will have a new thymine bond to it, and the same procedure will occur between any of the free cytosines and guanines. And of course, following the base pair rules, cytosine will pick up a new guanine, and a guanine will then, the free guanine will have cytosine bond to it. This process doesn't start on the ends. It will start to separate in many points at the same time. And if you realize a molecule that can be up to 3 meters long, containing billions of base pairs, this makes sense. It would take forever if it started at the ends and worked its way to the middle. And naturally, this process will only happen in cells that are going to divide. Any cell that is not going to divide will simply stop after the G1 growth period in the cell cycle, and it will not move to the S phase of the cell cycle. This way, the 2 new daughter cells that will result after cell division will receive their own full complement of identical DNA molecules. And now we'll begin to talk about the processes involved in protein synthesis. And here is where the different kinds of RNA will come into play. The first step is going to be transcription. During transcription, mRNA, or messenger RNA molecules, will move into the nucleus and will copy the info on the DNA base sequence. This happens very similar to how the DNA will unsplit or unzip and make a copy of itself during replication. The DNA molecule will unzip, the mRNA will come in and bond with the free nitrogen bases of the DNA molecule, thereby making a copy. And of course, since uracil is used by RNA in the place of thymine, instead of thymine bonding to adenine, we will have uracil bonding to adenine. Once this new molecule of messenger RNA is made, it will then leave the nucleus and travel into the cytoplasm of the cell. Once in the cytoplasm, it will then move over to the ribosomes, and of course we all remember that ribosomes are the site of protein synthesis. As this strand of messenger RNA moves into the ribosomes, it is now carrying the code for all of the proteins made in the body. And this code is written in a 3-letter word known as a codon. Each codon will code for a specific amino acid. And now the next step in the process of protein synthesis will begin, and it is known as translation. So now the mRNA with its 3-letter codon is in place in the ribosome. The tRNA will carry the amino acid that the mRNA codon calls for, and on the tRNA will be a 3-letter code that will be opposite of the codon, known as an anticodon. So as an example, if the mRNA codon is CAG, cytosine, adenine, guanine, then the tRNA anticodon will be the opposite, GUC, guanine, uracil, and cytosine. Each tRNA will bring its amino acid, called for by the mRNA codon, and drop it off at the ribosome, and the ribosome will shift, reading the next codon on the mRNA molecule and calling for the new tRNA anticodon carrying that specific amino acid for whatever the mRNA codon calls for. This process continues, linking all of these amino acids together. We know that amino acids are the basic building blocks of proteins, so we link all these amino acids together until the mRNA codon reads stop, and that indicates to the cell this protein has all of the amino acids it needs to be assembled. Since there are many, many different ribosomes all in the cytoplasm of the cell, both free-floating and attached to the endoplasmic reticulum, this process will be happening at many, many different sites found on the mRNA code, and it will all be occurring at the same time. So in review, each protein is made of one or more polypeptides, which will be made of a specific series of amino acids. The genetic code is built into the arrangement of the nitrogen bases in the DNA. The 3 nitrogen base codon in the mRNA will code for a certain amino acid and then will be read by the T or transfer RNA, and it will bring the appropriate amino acid that the mRNA code will code for. All of those amino acids will be linked together, and that is how we form a protein. And that will conclude tonight's episode of Mr. G's Sleepy Science. Tonight we talked about DNA structure and function and how it relates to replication, transcription, translation, and the importance of protein synthesis, all in front of a nice warm crackling fire. I've been your host, Mr. G, and once again, thank you for listening. Good night.